Good morning, students. Please inform to your friends. Let them join as early as possible. Within 10 minutes, the section will be started. Good morning, students. Please inform to your friends who had not joined. Let them join as early as possible. Within 5 or 6 minutes, the section will be start. Please be alerted. Okay. Good morning students to everyone. Within 3 minutes the section will be start. Please inform your friends who have not joined. Let them join as early as possible. YouTube live and Facebook live is also going on. You can check there also. Okay. Thank you. The topic is also different paths of drug discovery. Yesterday you had introduction and current throughput. Today different paths of drug discovery. Second day topic, day 2 topic. Please don't miss the sections because the very important topic is going on here. Students, please don't delay the class also. Please inform to your friends, let them join as early as possible. YouTube live and Facebook live is also will come. Will not have any disturbance in the network issue. Okay? Okay. All the best. Please join as early as
Maheshwari, can you hear me, Maheshwari? Maheshwari, can you hear me?
good morning, sir.
Yeah, very good morning. Just wanted to check with you, Maheshwari. When I use my cursor, Is there any way this pointer can be colored pointer, because lights are in the black white background? So this cursor, if it is colored, it will be helpful if at all you know.
Yeah, no, sir. Same it will come.
Okay, no worries, no problem. I'm there. Whenever you are ready, we'll start. Okay, all right,
Yeah, okay, sir.
no problem.
Students, last announcement. Please inform to your friends who had not joined. Let them join. Facebook Live and YouTube Live is also coming. Please check and you can join there also. Okay. And I am giving to you feedback form in the YouTube, Facebook also. Please check it. You are not at all checking and you are just typing. Please send me feedback link and you are calling to us. Please check. Be alert and check the link also. Okay. Okay, sir. Sorry for the delay. You can continue, sir.
We are ready to start.
Yes, sir. We
Okay,
can.
all right. Uh, just 1 second. Are you able to see my screen?
Yes, sir.
Okay, so we're ready to start, right?
Yes, sir. We can start.
Okay, all right. So very good morning to all of you, and welcome to skill development via e-learning initiative by Andhra Pradesh State Skill Development Corporation. We have commenced this program from yesterday, and this is, as you all know, it's a 15 days training on drug discovery and development and overview. So this is a day two of our training of our lecture series. So as we have discussed yesterday, we will actually be doing a quick recap on what we have learned yesterday, and then we will move on with our today's topic. Okay. So, as I said yesterday, we had talked about briefly about Life Science Sector Skill Development Council, about PVR research facility, because the whole drug discovery development is from a perspective of a research facility. I mean, from a research background. So, just wanted to share a few things about a contract research organization. And then we talked about introduction of drug discovery and development, where we have covered different phases of drug discovery and development, and how much time it takes, how much investment it takes, and what is the success rate. In brief, we have discussed about that. So we will have a quick recap of all those things, and then we'll move on to today's topic on alternatives to animal experimentation, because many a times, as you all know, we hear that animal experimentations are criticized because this testing is happening in live animals. So the question here is there any way that we can avoid an animal experimentation and still we can do our research? So we'll talk a little bit about of that, and then we'll talk about two cell-based models, especially hepatocytes, which is liver cells, and enterocytes, which are intestinal cells, as tools for metabolism and toxicity studies early in drug discovery. So that's the overview of what we're going to talk or cover in today's talk. Okay. So yesterday we just started with our course content discussion. So again, to re-emphasize, we will be covering a current scenario, different phases of drug discovery, structure-based drug designing, QSAR and computer-aided drug designing, in vitro screening, in vivo screening, DMPK studies, efficacy studies, clinical trials, high throughput screening and early drug discovery, and a role of different disciplines like chemistry, microbiology, pharmacy, biotechnology, and research. So this is the whole course content which we are going to cover in next 13 odd classes. Okay, we started yesterday with introduction and current scenario to drug discovery and development. So we discussed about License Sector Skill Development Council, a non-profit society, as an initiative from Ministry of Skill Development and Entrepreneurship, and the sole aim or the mission of License Sector Skill Development is skill development. Okay, India being one of the important country from perspective of largest exporter of vaccines, we are one of the largest biopharmaceutical sector in the world. Like as we discussed yesterday, more than 60% of vaccine demand from the rest of the world is met by Indian companies only, and more than 50% of World Health Organization and UN requirement of vaccine is met by Indian companies. And who are the major manufacturer? Serum Institute. We have Bharat Biotech, Indian Immunologicals, and many other companies, like including Zydus and all, who are into vaccine manufacturing. And about the council, it is it comes under NSDC, National Skill Development Corporation, under the mandate of Ministry of Skill Development and Entrepreneurship. So far, we have done quite a few many placement for the candidates, because it's a government initiative. Around 20,000 candidates have been placed so far. Training partners across country, more than 300 are there. We have more than 700, close to 700 industries from life science sector aligned with LSSDC. Okay. Within life sciences sector, pharmaceuticals, biopharmaceuticals, and contract research organization. All these areas comes within the purview of Life Science Sector Skill Development Council. India, as I was mentioning, that one of the important biopharmaceutical sector, but also it is the third largest pharmaceutical industry in the world by volume, and including Dr. Reddy's, Sun Pharma, Zydus, Torrent, Intas, Lupin, Orbindo, Mylan, Teva, Granules. These are all the major players in pharmaceutical industry within India. Council also works in close collaboration with government and ministry departments like CDSCO, Central Drugs Standard Control Organization, Department of Pharmaceuticals, Ministry of Chemical and Pharmaceuticals, and Department of Biotechnology. Within the governing board members, all the major associations, manufacturing associations, contract research associations, or pharmaceutical alliance, IPA and OPPI, IDMA, all of them are on the governing board. And the chairman of the council is Dr. Satish Reddy, who is chairman and managing director of Dr. Reddy's Laboratory. One important slide we shared yesterday about the employment snapshot for life science sector. So more than 50% job employment opportunities lies with manufacturing, around 10% with research and development, around 24% in supply chain, and around 4% in 24% in marketing sales, and around 4% in supply chain and logistics and other departments. By qualification, there is a scope for, I would say, all the levels of NSQF levels, like you can call it as from 10th, 12th pass to ITIs, diploma, graduates, postgraduate, PhDs. Everyone has a role to play in license sector.
Can you increase
8% job roles are.
Excuse me, sir. Can you increase your
Yes,
brightness in your laptop?
Maheshwari, please go ahead.
Can you increase the brightness in your laptop?
It is breaking. Can you just speak again?
Can you increase your brightness in the
Maheshwari?
laptop?
Oh, increase the brightness on the laptop?
Yeah.
Okay. 1 second. Is it any better? Maheshwari, is it any better?
Yes, sir. You can continue.
Okay, great. Okay, whenever you feel like you can come back to me and you can let me know. Okay?
Yeah, okay.
All right. So yes, so also there is an apprenticeship program as an initiative from ministry as a special scheme called National Apprentice Promotion Scheme. So if students register for this, they will have an opportunity to be picked up by local industries, pharmaceutical industries, as apprentices, and then after their performance review, they can be on their payroll as a permanent employees going forward. So up till now, in last year, couple of years, we have done more than 15,000 apprentices in different industries across country. These are the major job roles which are from the license sector in different industries. I just enlisted them. We are also running some e-certification programs like pharma production, medical representative, quality analytics. These are 1 month to 1.5 month long webinar programs. We also run an initiative called e-Pathshala, which is 1 week accelerated programs where so far we have started with drug discovery and development, analytical technologies, and going forward we'll have some more job roles like microbiology, QC microbiology, and across country. Uh, and then we have from Telangana, we have Swarna Bharat Trust as one of our center of excellence. This is just a logo of e-Pathshala initiative of the council. This is the program details about few programs which are running through e-Pathshala. A contract research organization, which is again our only mission is to provide research services to different sectors within life science sector. So, what are the capabilities or expertise? It is in analytical, bioanalytical, microbiology, biotechnology, pre-formulation, DMPK, pharmacology, toxicology, research and training. And industries we support are pharmaceutical, medical device, cosmetics, biotech, agro-food, food supplement, feed additives, and herbal and chemical industries. These are just the snapshots from the lab, how it looks like. There is a CPCSA approved state-of-art animal research facility also. So these are a few snapshots from that. Then we also talked about one of the product as an enzyme, like because we are talking about drug discovery and development. So Cariolap is one such product, an enzyme mixture which was launched in 2016, and the background was like for a veterinary application in milking animals, only 50% feedstuff gets digested, and another 50% comes out of their body undigested in feces. So somehow, if we can improve the digestion, then overall efficiency of milking animal can increase. So it is that product, and this 9 enzymes in total. It covers. They are responsible for digestion of more than 90% of the feedstuff which are available across country. Okay, this is officially it was launched at a government function. A few snapshots. It was duly covered in media because it's a novel approach where no drugs, no hormones, nothing is used. Simply by improving the digestion of milking animals, their milk yield or their fat percentage can increase. Um, Another just a novel application. I won't say novel discovery, but a novel application. UVC light, which are at 250 nanometer, very portable device. Where in current scenario of COVID, if you can use this and expose all your daily use items or test articles, they can in this box. So this is a very affordable, portable, easy to use UV disinfection box, which utilizes the principles of UVC light, which emits at 254 nanometers. Also, a lot of dissertation work, 1 month, 2 months dissertation work, research work goes on here. We tie up with a lot of academia and help their PhD students also to select novel research program and write a thesis out of it. And even we help them in doing article publication. And every year we do this summer training along with biotechnology department. So these are a few snapshots from the trainings that have happened. And then we jumped into our topic that was drug discovery and development. So yesterday we started with the introduction part. So as we have discussed a roller coaster ride, so same way drug discovery development field is highly uncertain. So you never know whether how much time it will take, uh, whether it will succeed or not. So there are so many uncertainties, as is the case with roller coaster. But same in a roller coaster ride, we feel so happy, we feel so excited, and we scream out of joy. Same is the case with drug discovery and development. Every single step is interesting, full of uncertainty. But yes, with every step, whether a product can go forward or it can fail or it can succeed or it can cross that step, there is a lot of learning for chemists, for biologists, for analytical chemists, for biotechnologists, microbiologists. There is a scope for a lot of learning into it. So that's why we call it drug discovery and development as a roller coaster ride kind of thing. A few cartoons we just shared yesterday, like a drug is taken in the buttock, but how does it reaches and elicit its action for a sore throat, right? Again, there was a time in history we know few many drugs have come into market by chance. I mean, they were discovered by chance. Biggest example is penicillin. So that way also drugs can be discovered. Also, it's a fact that many a times the side effect to the drug is more than the disease itself. So that's also one objective for drug discovery development scientists, which keeps us motivated that yes, we should really come up with safe and efficacious drug, so that safe in the sense there should be as minimal side effect as possible, and efficacious means it should be highly efficacious. So that's the whole goal for drug discovery and development. So we talked about the process yesterday. The drug discovery development process can be divided into different phases, namely discovery, early discovery, preclinical, clinical, and then it finally goes for approval. So this is a long process. It takes around 10 to 15 years of time to come up with one successful drug molecule into the market, and it takes huge investment to the tunes of $2 billion. So it's a very costly affair. And what is the success rate? And success rate, unfortunately, is only 0.003%. What does it mean? That out of 30,000 molecules which you have started your project with, okay, maybe at early screening stage of in silico or chemical synthesis stage, eventually when you come up with only one molecule in the market. So you see 1 by 30,000 is just 0.003% success rate. How do we arrive at this 1 by 30,000 as 0.003%? So we have learned yesterday that around 2,000 molecules, around 7% of the molecules will only enter into preclinical development. Out of those, hardly 200 will enter into clinical phase trial 1. Only 40 will get into phase 2 clinical trials, and hardly 12 or less than 12 will get into phase 3 clinical trials. And out of the drugs which are actually approved by the regulatory agency, only one drug will give you the return on investment. So that's why we say the success rate is 0.003%. So we talked about different phases. In each of the phases, what are the steps involved? So we started with early drug discovery, where the first step, as you may understand, is target identification. You need to identify a disease which you want to treat. So that is your first step in drug discovery development project. And then you should have a target, which mostly it could be a molecular target, it could be an enzyme, it could be a cell type or pathways. So it can be a molecular target which will bring about a treatment or cure of your disease. So that target you have to identify, and only identification doesn't help. You have to actually validate that target by doing several in silico modeling and make sure that that particular molecular target interventions will actually help you in treatment of the disease. So you have to validate that target, and then we do the hit discovery where a lot of molecules will be screened against that target. And then when you actually want to do an initial screening, you should have an in vitro assay in place. Okay, so you have to develop an assay where that molecular target can be expressed, and either the pathways can be inhibited or it can be induced, whatever is your mechanism based on the disease. You should be able to reproduce that in a model, in a cell based or an enzyme based model. So you have to develop an assay for that. So more and more molecules can be screened with that. And as I discussed yesterday, we will be doing high throughput screening also. We'll be covering within this scope of this training. And high throughput screening means most of the things we do it in high throughput fashion, so they can be done in a larger scale, in a quicker fashion. And in today's scenario, when companies are working on so many targets, so many diseases, it is very important that they should have STS platforms with them at different stages of drug discovery, be it a hit or lead finding stage, chemical synthesis stage, or doing a biological assay, or doing an in vivo assay. Every stage, if you have STS involved, then the whole process can be slightly quicker. So even bioanalysis can happen in high throughput fashion. So we will cover that also within the scope of this 2 weeks of training. And then hit to lead. Finally, you have one lead molecule or a scaffold which really works well for you, a chemically synthesized drug. And then within that scaffold, you will do some optimization. So that is called lead optimization. In preclinical, we have learned that either there will be in vitro assays, which would be cell-based or an enzyme-based assays, or there will be in vivo assay, which will be testing in animals. Animals means there will be laboratory animals. It could be rats, mice, guinea pig, hamster, beagle dogs, anything but rabbits. They'll all be laboratory animals where we can test it. And then we will be doing ADME studies, absorption, distribution, metabolism, excretion studies. We will also be doing proof of concept studies. Finally, you have to study and optimize the drug delivery, which is very important because it is equally important than to have a safe and efficacious drug. It is equally important the delivery should be very optimal. And formulation optimization takes place. Finally, with all this data, we file our application for IND, which is investigational new drug application with the regulatory agency, generally with FDA. And they will review that data, and based on that data, they will approve the clinical trials. They will give a permission that yes, your data is sufficient enough. Now, same molecule you can go and test in human. So that comes under clinical development. So there, very importantly for you guys to remember, just 3 to 4 phases. Remember, phase one clinical trial, which always happens in healthy volunteers. Okay, and then we do in phase two, which is patients population. In phase three is also in patients. Number of volunteers in phase one varies around 20 to 100, from 100 to 500 in phase two, and 5 and thousands numbers would be there in phase three. So we do all dose escalation, single ascending, multiple dose studies, safety, efficacy, DCGI, or any other country regulatory agency where we want to register our drug. And once it is approved from
Sorry,
them,
sir.
finally,
Sorry for announcing.
yes,
Your screen is not visible
it's not visible. This is so strange.
just now. You lost your network, but that it is not visible. Sir, can you connect the
I,
land cable?
yes, exactly. I was just about to do that. Can you just give me one moment, please? Yeah, I'll just do it immediately.
Yeah.
Are you able to see now, Maheshwari?
No, sir. Present your network also is low. Red
Yeah.
signal
Can you
is
just see
showing.
now? Is it better?
No, sir. It is
Maheshwari?
blurry your face. Yeah, your video also not visible. Clarity.
Oh, one second. One second. I'll do something about it.
Yeah, now it is okay.
Okay. Is it visible now?
Please share your screen, sir. Screen is not shared.
I'm so sorry. Okay.
Yeah, not an issue, sir.
Yeah. Is it visible now?
Yeah, yes, sir.
All right.
It is
I'm so
blind.
sorry about the.
It's
Go ahead,
blind.
please.
Fully
Can you
black
see
screen.
Maheshwari?
That's all. Your screen is showing as black screen, sir.
This is so strange.
PPT is not showing.
One second. You'll have to give me just few more seconds to fix this. Huh? Just.
Yeah, okay. Sorry, students. Sorry for the interruption at between the class because we are having some network issue. It will continue. Students, please check. I have given link to YouTube and Facebook and Airmeet in the YouTube and Facebook also I have posted just now. You can check and you can join. I have given Airmeet, YouTube and Facebook links in I have posted in to everyone. Please check it. I have posted in YouTube and Facebook also just now. Webinar links you can join. Sorry for the interruption. Sir will join within seconds. Yesterday feedback also I have given to you students. Please check it. Again I will post today in YouTube, Facebook and Airmeet. Please check it and give the feedback thanks.
Maheshwari, can you just once check and confirm it is fine because the IT person is working on that. He will fix it. But meanwhile, can you just tell me now is it visible?
You are visible sir, but PPT is not visible sir.
Yeah, yeah. PPT only. I'm saying PPT now. I have shared actually.
It is blind.
I don't know why it is happening. One second.
Once close the screen share and again once open PPT sir. Yeah, okay. It is visible. You can continue.
Is it visible now?
Yes
Okay.
sir.
All right. So maybe I closed and opened it again, so it must be working. Okay, great. Let's continue. And still, if you feel some issues, you can let me know because IT is working on the LAN connection. They will
Yeah,
fix it.
okay.
Before that, I've just taken it on a separate hotspot. Okay. All right. So my sincere apologies for slight hiccup because of technical glitch. So all right. So we were talking about post-marketing surveillance on this slide. So once the drug is launched into the market, you will have a post-marketing surveillance happening, and that we call it as phase 4 clinical trials. Yeah. So in summary, we talked that you should have a disease identified first for which you want to develop a drug. Identify the molecular target. Once you identify that, you need to validate that target. Finally, do your lead optimization. So a lot of molecules in silico from your library you can screen. Those would be called as hits. From that, the one where you find maximum efficacy or the characteristics of good binding and all those things, that you select as your lead. And then further we do the lead optimization. Finally, in preclinical, after doing all the in vitro cell-based, enzyme-based assays, you can test them in the animals, which will be called as preclinicals. And then that data, once we submit as an IND application, if it is approved, then you go for clinical trials in human. There we do phase 1, 2, 3. Then we submit our application for approval. The drug is launched in the market and it can be used for the disease treatment. So this is the nutshell flow of drug discovery and development. We talked about
Excuse me, sir.
yes.
Sorry, your PPT is not visible again.
Okay, so then just give me a few seconds. I'll let IT fix it immediately. Okay,
Yeah.
thank you.
Sir, please on your mic.
Yeah, Maheshwari, just working on that. Just give me a few more seconds. Sorry about that.
Yeah, okay. సుచిత్ర టైల్మెట్ లింక్ అనేది మీకు ఫోన్ లో ఓపెన్ అవ్వదమ్మా. అందుకనే మేము లైవ్ ఇస్తున్నాం, యూట్యూబ్ లైవ్ అండ్ ఫేస్బుక్ లైవ్. ఓకేనా? నేను ఆల్రెడీ ఫీడ్‌బ్యాక్ లింక్ యెస్టర్‌డే పోస్ట్ చేశాను. ఇవ్వలేదని మాత్రం నన్ను బ్లేమ్ చేయొద్దు. ఓకే. వై యూ ఆర్ డూయింగ్ బ్లేమ్ టు మీ? ఎన్నిసార్లు పోస్ట్ చేశాను? థ్యాంక్ యూ తేజస్వి ఫర్ పోస్టింగ్ ద ఫీడ్‌బ్యాక్ లింక్ ఆన్ యెస్టర్‌డే. రియల్లీ ఐ థ్యాంక్ యూ. ఎందుకు మీరు అలర్ట్గా అట్లా ఎక్కడో పెడుతున్నారు, ఏదో వస్తున్నారు, ఏదో వెళ్తున్నట్టు చెయ్యొద్దు ప్లీజ్. సిన్సియర్ గా నేర్చుకోండి, సిన్సియర్ గా చూడండి. ఫీడ్బ్యాక్ లింక్ నేను పోస్ట్ చేయలేదా? చేయనప్పుడు తేజస్వికి ఎలా వచ్చిందో చెప్పండి. తనకి నేను ఏమన్నా పర్సనల్ గా చేశానా అడగండి. మీరు చూసుకోకుండా ఎవరిని అంటారమ్మా మీరు? ఇన్ ఎఫ్బి అండ్ శ్రీవల్లి, ఎఫ్బిలోనే కాదమ్మా, యూట్యూబ్ లో కూడా పెట్టాను, ఒకసారి చూడండి. అదిగో ఎఫ్బిలో వచ్చిద్దా అమ్మా? యూట్యూబ్ లో మీకు ఎవరికి ఫీడ్బ్యాక్ లింక్ రాలేదా చెప్పండి? ఎనీవన్ రెస్పాండ్ హూ హ్యాడ్ గివెన్ ద ఫీడ్బ్యాక్ లింక్? లేదా నేను ఇప్పుడు పేర్లు చదువుతాను, ఇచ్చిన వాళ్ళ పేర్లన్నీ చదువుతా వరుసగా. యూట్యూబ్ లో మీకు ఎవరికి ఫీడ్బ్యాక్ లింక్ రాలేదా? ప్లీజ్ రెస్పాండ్ ఎనీవన్. నోనా, నాట్ మౌనిక, నువ్వు ఒక్కదానివే కాదు, ఇంకా ఉన్నారు, నీకు రాలేదని చెప్పకు. ఇంకా ఇచ్చిన వాళ్ళు చెప్పండమ్మా, లేదంటే నేను ఇక్కడ ఎవరెవరైతే ఫీడ్బ్యాక్ ఫామ్స్ ఇచ్చారో వాళ్ళ పేర్లు చదువుతాను, వాళ్ళు చెప్పండి. అంజని, హారిక, లావణ్య, షర్మిల, హర్షిత, ఉదయ్ భాను, చెప్పండమ్మా. అదిగో వచ్చిందంట. మీరు చూసుకోకుండా రాలేదు, నో మేడం, ఏదో రాలేదని ఒక్కళ్ళే పెట్టేస్తే సరిపోదమ్మా. రాలేదు మేడం, యూట్యూబ్ లో రాలేదా? ఒకసారి చూడండి అమ్మా. ఇవాళది కాదు నేను చెప్పేది, నిన్న యూట్యూబ్ లో మీకు ఫీడ్బ్యాక్ ఫామ్ ఇవ్వలేదా? నిన్న సంగతి అడిగేది, ఇవాళది కాదు. యూట్యూబ్ అండ్ ఫేస్బుక్ లో కూడా ఇచ్చానమ్మా, యూట్యూబ్ లో కూడా ఇచ్చాను. యూట్యూబ్ లో నుంచి ఎవరైతే చూసి ఫీడ్బ్యాక్ లింక్ ఇచ్చారో వాళ్ళు రెస్పాండ్ అవ్వండి అమ్మా ప్లీజ్. వచ్చింది, మీ నెట్వర్క్ ఇష్యూ లేకపోతే అది అవ్వకపోతే నేనేం చేయను చెప్పండి? మెయిల్ కి పెట్టాలా? ఇంక రోజు మీకు మెయిల్స్ పెడుతుంటే నేను ఆఫీస్ లో ఏం చేయాలి? టుడే ఇస్ ద లాస్ట్ డే. నిన్నటిది వాళ్ళది కలిపి మళ్ళీ వాళ్ళని పోస్ట్ చేస్తాను. మళ్ళీ మీరు రిపీట్ చేస్తే నేనైతే ఫీడ్బ్యాక్ లింక్ ఇవ్వను. ఇచ్చిన వాళ్ళందరూ పిచ్చోళ్ళ? నో మేడం, నో మేడం అంటున్నారు. సరే, వాళ్ళు చూసుకోకపోతే నా తప్పేం లేదు. సారీ ఫర్ ద ఇంటరాక్షన్. సార్ విల్ జాయిన్. ఐ విల్ కాల్ టు సార్ వన్స్ అగైన్.
I lost my link also. Your internet is unstable. You see this? This is an internet problem only, I see. I'll continue with this.
Sir, are you ready to continue?
Yes, Maheshwari, if you can just display the presentation.
Yeah, okay, sir.
Yeah. Yes, that's correct. All right. I'm so sorry for the technical glitch, some internet issues, but okay. So, so now your slides are visible, right, Maheshwari?
Yes, sir, you can continue. Yes, sir, you can continue. You can see my PPT.
Yeah, if you can just make it full screen, yes, great. So, all right. So we just talked about the drug discovery development phase-wise process. On the next slide, we this is the whole summary from target validation identification to all the way to approval and launch into the market. On the next slide, uh, we we also then talked about the role of different fields, different disciplines at different stages of drug discovery. So we have seen the role of microbiologists, biotechnologists very early initially at target selection validation, because basically you're talking of genomics, proteomics, bioinformatics. So your knowledge of molecular biology will really help. And then when you do the initial synthesis of your molecules for screening, your chemistry background with organic or inorganic chemistry background really helps. While doing the biological assays, your pharmacy background really helps if you want to do cell-based, enzyme-based assays, or you want to do in vitro experiments with cell-based assays. Your microbiology background comes into the play, and if you are from zoology or veterinary background, then when you do animal studies, testing of this drug in animals, there actually it helps. So there is a that's why we say that it's an interdisciplinary function, drug development, where chemistry, biotechnology, microbiology, pharmacy, biotechnology, analytical chemistry, organic chemistry, inorganic chemistry, even statistics, biostatistics, all these fields play a role. On the next slide, uh, then we uh, then we also see the difference, be it a small molecule or large molecule. The drug discovery development process is same. but there are few changes because of the difference in the approach. Say because biologics are more complex compared to chemically synthesized small molecule. Just by an example, we have seen acetyl salicylic acid, just a small molecule, hardly 0.18 kilodaltons of molecular weight. On the next slide, apart from the molecular weight difference, uh, can you go to the next slide, Mahesh? Yeah. So we have seen that even post-transitional changes are also there. And if you see a cycle of biologics, how they are basically manufactured, then you will see that the basic difference is chemically synthesized one are small molecules, whereas biologics they are made from living. So almost every time the cycle starts from transfection of DNA into your host cell. Then you go for appropriate cell line selection, and then you further develop that, and then you would also like to have a cell banks where all these things can be maintained. Further, you go for fermentation for a large scale thing. Finally, you would like to isolate, do the purification, characterization, and the stability of your molecule, large molecule. And then once the formulation is developed, you would package it, do the stability studies or refrigeration storage happens, cold chain, and then finally it comes to the treatment as a for the patients. On the next slide, uh, this is in general we have talked about biologics. So there is a difference between a biologics versus a small molecule. So what is this difference? A chemically synthesized molecule always follow some same strict protocol which is being developed. In other words, once your reactions are validated, then almost every time you can get the same yield, you can get the same compound exactly same as long as your process is same, right? So it's sometimes it's very easy to replicate because you have standardized your process, the reactions, chemical reactions for synthesis of molecule. Whereas for biologics, since there is a living cell involved, there are so many steps for the transfection and then cell lines you do, and then you do fermentation. So at any of these steps, if there is a small change, can lead to change in your yield. So that's the major difference why we call biologics a little more complex, not only from the structure but also for manufacturing purpose compared to small molecule chemically synthesized drugs. On the next slide, so typically, when a biologics is submitted for IND application, we have seen this is an example taken for cancer biologics. So generally, the questions asked by regulatory agency are same, like they would like to know the pharmacodynamics, okay, they would like to know the pharmacokinetics, and the toxicity or the safety profile of your drug. So that's pretty much same as it is required for small molecules also. On the next slide, then we also talked about. Can you just go to the next slide, Maheshwari? Maheshwari, are you there? All right. So on the next slide, as you have seen, this is a drug discovery and development funnel. So basically, why do we call it as funnel? Is because funnel typically is having a wide entrance, whereas as you go down, the it becomes narrower. So same is the case. We start with many molecules, even at in silico stage or during early drug discovery. But by the time you actually are at a stage of launching your molecule into the market. It's only one or two molecules which makes it successfully to the market. So that's why we call it as funnel. You start with thousands of molecules. After initial drug discovery screening, you will go to the preclinical stage where again a lot of screening happens. Molecules will be screened, and then with very fewer molecules gets into the clinical trial, and after completing 1, 2, and 3, and then phase 4 stage, only one or two molecules will reach. In total, more than 15 years, 12 to 15 years of time, and billions of dollars of investment. So that's why we call it a slow and a very costly process. On the next slide is about the cost, because why should we on the next slide? So there we have talked about the cost, because this is just a survey I have taken from a literature from 1995 all the way to 2014. Every year the cost of R&D has increased. Okay, so from $15 billion to 2014, now it is even double of that. But by 2014, it was $51 billion of investment. So in a way, it is good because investment is increasing. But at the same time, unfortunately, the success rate is not increasing. You don't have that many molecules coming into the market as the cost of R&D is increasing. But it's good. Why I'm saying is because all of you are life science sector students in pharmacy or microbiology or biology or zoology or chemistry, or all of you guys have real good scope because there is huge and huge investment getting into R&Ds. On the next slide. So now all of you know, we have discussed yesterday that at least we know that why we say that drug discovery is costlier, right? Because many times the single strip of drug will cost you huge. So why it is that much costly? Because more than $300 million of investment goes into the preclinical phase, more than $400 million of investment gets into clinical, and even after drug is launched into the market, more than $100 million investment happens there. And that's why the drug discovery development is costlier, and sometimes we see the pill or the capsule or a tablet which is coming out in the market are actually costlier products. On the next slide. So now we talked about the costlier thing, why it is costly. Let's understand why it is costly. So the whole cost is not only the cost of a successful drug, but the cost is also incurring on the drugs which fail. Because you start with thousands of molecules, it requires investment. Not all thousand will make it to the market, as we have seen, only one. But you have to do screening. So it is the cost incurred by the companies for investment on failed drugs. That also adds up to the overall cost. Okay. On the next slide. So we say fail early, fail cheap. So a drug which fails in preclinical phase is cheaper than a drug which fails after clinical trial. So that's why we say that do stringent screening. You should have all those liability studies, safety, toxicity studies done very initially during early drug discovery, so that a drug which has to fail will fail. So chances of failure of a drug going forward would be less. So these are a few reasons why we discussed yesterday why a drug has to fail. So more than 40% reason because of poor pharmacokinetic profile, more than 20% reason because of toxicity, more than 30% drugs have failed because of lack of efficacy. So this is an old slide, but now there is an improvement. Now it's not 41%, but this failure because of poor PK properties has come down to less than 10%. And the reason being the advancement in technology and our understanding of pharmacokinetics. On the next slide, if you see here, we are seeing the reasons why a particular drug really fails. So just for your quick understanding, for preclinical reasons, it is majorly toxicity. For phase 1, 2, 3, For phase 1, it is of course safety, but phase 2 and 3 is always lack of efficacy, because of which drug can fail. So on the next slide, this is just a summary of what all things we have discussed. Like I have taken an example of Pfizer, hundreds of discovery approaches they do. And if you see on that slide, it's again a discovery funnel. You can see where hundreds of molecules are taken up, thousands of molecules. Here it's written as millions of compounds are screened, they go to preclinical, from there they go to clinical, and then from clinical, finally only one or two will make it to the market. So even the companies like Pfizer, which are the world's top 3 pharmaceutical companies, they also have to undergo the same process of drug discovery and development. Many years, 11 to 15 years, and more than 800 million or $1 billion of investment takes into this. And the success rate, hardly one or two product comes into the market. On the next slide, last slide, yesterday's discussion, we talked about the drugs which are withdrawn from the market. Okay, so what we have discussed is the whole process takes around 15 years, but still, after doing so much of research, so much of understanding on every aspect of the drug which is possible, still when a drug is launched into the market, there are chances that drug can be withdrawn back from the market. Why? Because of the reasons enlisted over here. On the last column, if you can see, because of hepatotoxicity, because of lack of efficacy, because of lack of clinical improvement or clinical benefits, because of cardiac toxicity, because of liver damage. So you see, these are the major reasons. So what I'm trying to convey through this slide is that no matter how much research, how much understanding is there for the whole process of drug discovery and development, still there is some chances that there could be some unknown, some hidden side effects which could only be mapped after the drug is launched into the market. Okay, although with every year, with our improved understanding on these processes and improvement in the technology, the process is going to be more and more safer. But as a student, we should know that still drugs are being withdrawn from the market. This is just a summary of all the discussions which we did yesterday, starting from early drug discovery, preclinical screening, clinical studies, and finally approval from the drug. So that's about the review of our yesterday's thing. Okay, and then on the next slide is like we didn't really cover this in yesterday's discussion, but I will spend at least a minute on this slide because this will really make you understand what all things are important in drug discovery and development. So as we have understood that, you see, target, you need to have a disease target in place at the first place, okay, to start with. Once you have a target, we really want that our drug should be targeted. In other words, if there are any off target effect, okay, any molecular mechanism you're trying to target, if our drug will have an impact on any adjacent target, it can lead to lot of toxicity, lot of untowards effect. So we want that our drug should always be targeted. That's why we say that it should be highly selective. Okay, it should be highly selective to the molecular target we are working on. Only then you will have maximum efficacy and least chances of having side effects. Because you see, in our body, physiologically at a molecular level, you can see it's a complex mechanism, right? So when you try to capture any particular pathway, then you have to be very sure that only that pathway is we are doing some intervention. If at all you try to affect any other adjacent pathway or a dependent pathway, then there could be side effects. So it should be highly targeted, it should be highly selective. At the same time, it should be potent, right? I'll tell you, when I started my career, there was a time when we even have worked on drugs where the potency used to be micromolar, right? But now with this so much of advancement in our understanding and our focused efforts in chemistry and biology, it is possible to bring to come up with highly efficacious drugs. So we are talking of potency in micromolars or even nanomolars, picomolars level. So it should be highly potent. It should be stable. Of course, it should be stable. If it is not stable, then again, that is not advisable. So whatever physical form of the drug is there, we want that it should be stable. It should be permeable, and of course stability, this is physical stability of formulation I'm talking about, but it also means that when you actually give this drug goes to the liver there, what is the stability, because based on that stability the half life and how long a drug would be efficacious will stay in your body, it will be decided. Okay, it should be soluble, because unfortunately we will be covering in subsequent talks about BCS classification of solubility. So there is class 1, 2, 3, 4 types of molecules under research, and unfortunately we always have to deal with compounds which are very low soluble and low permeable. So if to start with, if a chemist can make the molecules soluble, it will really help for the pathway of the drug. Permeable, it should be permeable, because if it is not, you can go back to the same slide. If it is not permeable, then it cannot pass through the blood brain barrier. So that's the reason it should be permeable also. So if you see, we always say that if a drug is not permeable, of course there are means and ways where we can make this drug permeable, but it's always advisable to address this permeability issue very early in drug discovery. Okay, if it is later, then you will have to put in that much more effort. So we always suggest that at least permeability and all those things should be addressed very early itself. Then metabolic is stable. We already have talked about non-inducing, reversible, durable. So this is all about drugs we are talking about. This is debatable. Like not necessarily every time the binding to the target, the ligand binding should be reversible. It depends on what target you are dealing with. When you talk of durable, again it depends on the target disease. Suppose it's a diabetic or a neurological disorder, we want the drug should be long acting, right? But if it's an antibiotic, we want that drug should be quick. So it should elicit the effect and it should come out of the body also at the earlier. So based on the target which you are trying to fix, it depends whether it has to be durable, the binding should be reversible or not. It should be non-teratogenic. By all means, the drug, from the safety perspective, we always want that drug should be non-teratogenic. Means it should not lead to any congenital abnormalities, right? So it should not have that potential for being teratogenic. Non-mutagenic also, it's also related to the safety. So this is also desirable. Finally, it should be patentable. So if you see, if you work so much on your drug and if you haven't looked at the patent possibility, and after putting in so much of time, resource, energy into this project on a molecule, if you come to know that, oh, somebody has already applied for the patent for this kind of a structure, then all your effort will go waste. So you should actually see that the target, the drug which you are trying to synthesize or discover, is having patentability opportunity also. Finally, it should be manufacturable. So you see, so many criterias are there. So only then, if your drug, if your molecule can pass through successfully through all these criterias, only then it can be successful. So now you see why we say that you start with hundred thousands of molecules, but end result is only one molecule successfully launched in market, because it has to pass through all the criteria. It has to meet all these criteria. Same way on the right side, if you see, this is exactly, we have talked about the studies, like all the discovery criteria are met, whether it is safe, whether your formulation is really optimal. Do you have the stability data? Some student has asked yesterday about the stability. So yes, do we have the stability of this data in place, of this drug in place? Then do you have all the trial things worked out, where you want to do the trial, multicentric trials, who are going to be the investigators, how how you're going to do the patient recruitment when actually you do the clinical trial, what is going to be the dose range of that, if there are any side effects, are you able to profile those side effects, have you done the long-term safety studies for this, have you planned carcinogenicity studies, is it manufacturing is fine, but is it really cost effective manufacturing, right? And then if there are any competitors, then do you have that competitive edge on your molecule? And finally, do you have a plan in place for regulatory agency filing of your molecule, the data for that molecule? So you see, this is a whole long process, and that's why you start like a funnel that is hundreds of molecules, and by the time you have one molecule successfully launched in the market, it is 10 to 12 years of time, and only one can make it. So this is, I hope now the students will understand why we say it is 10 to 12 years or 12 to 15 years of process of drug discovery and development, why so long time it takes. On the next slide, if you see, this is just a snapshot summary again, that once your target validation identification is done, you, even while validation, you can do an in vitro assay where you can test on a cell based or an enzyme based platform. Then you can do animal testing in vivo at a preclinical stage for a lead identification. I was talking about thousands of molecules. So earlier was the time when actually we used to synthesize many molecules, but now there are compound libraries which are available. All these structures are available, softwares are there. Each companies are having their own compound libraries from different programs. So those are ready-made libraries available. So you can really screen your target against all those available library compounds, right? And you can get the hit out of it. Correct. Again, the other way is from a literature, published literature. Also, you can get some compound ideas. And one of the most widely followed ways still from the natural products. Right. We can screen natural products and we can find some active pharmaceutical ingredient out of that. And finally, rational drug designing. We'll talk about it in detail in a separate lecture where we'll see the computer-aided drug designing, rational drug designing, how does it happen, what is the chemistry which gets into that. Okay. Finally, you have to do a proof of concept study. Friends, this is very important because you need to have a disease model in place, and only then you can do the testing in that disease model and prove that it could be a knock-in or a knockout, knockout animals, laboratory animals, and you can validate that the target which you have chosen for the intervention of disease is correct. Right. On the next slide. So this is, I think, in nutshell about introduction of drug discovery and development. Now let's talk about cell-based models today. Okay. So all the pharmacy students, it will be useful. This is the most recent. Of course, the cell-based models are there since long, but I'm also going to talk about a model which is most recent. So if you can learn about it while you're in college, in academia, it will be helpful when you get into industry, or even when you want to do your dissertation work or research work. This would help you there. So we'll be talking about alternatives to animal experimentation, and models I was talking about are hepatocytes and enterocytes. On the next slide. If you see, the question here is about animal experimentation. Okay, so Maheshwari, can you go to the next slide? So animal experimentation, all of you are science students, you'll be knowing that one of the important phase in drug discovery and development is animal experimentation. Okay, so what is animal experimentation? Is once you do your in silico screening using computers, once you do your in vitro assays in your lab scale, either a cell based or an enzyme based assay, finally, you would like to test the efficacy or a safe drug in a live system. Now, you cannot directly test in human. If that is the case, then imagine how many trials. So there is which is laboratory animal testing. So what we do, we do all this testing in animals. But with earlier, I'll tell you, I'll be very honest that there was a time when animals were used indiscriminately. Okay, like they were like just like an in vitro assay. For every molecule the chemist synthesizes, as a biologist, we just used to take the animals, dose to them, and then collect the blood, and then used to look for the PK profile, look for the efficacy and all, and go on. So what was happening was there was indiscriminate usage of animals across globe. But I'm glad that with time, scientists have also become aware. Animal ethics and welfare practices also came into play. In India, the regulatory body is called CPCSEA, okay, which comes in the Ministry of Environment, Forest. So CPCSEA, their only job is to ensure the ethics, animal ethics and welfare in research. So all the organizations, all the academic institutes who are having animal house, or the contract research organizations, or the industry who are having animal testing facility, it needs to be registered under CPCSEA. Okay, and there are certain guidelines. You can Google search. Even I will share. There's a CPCSEA guideline document. A small book is there where all the welfare, ethics, scientific, technical, and animal housing and all the details have been mentioned. I would want that all of you should go through, so at least it will help you to be a better scientist. Okay, as far as animal usage is concerned. So that's about animal experimentation. So now these are the few questions which people always ask: is it really required? Okay, it is debatable, but if personally, if I have to answer this question, my answer is yes. Animal experimentation is required. Okay, are there any better alternatives? Again, my answer is yes. Of course, there are better alternatives. Okay, so the third question is drug discovery. Is it really possible without animal experimentation? Then my answer is unfortunately no, because I don't think worldwide, it's not only about India, worldwide I would say we are not yet reached a stage where we can completely bypass this animal experimentation. Yes, of course, there are alternatives. We can limit or we can reduce the use of indiscriminate usage of animals. That is completely doable. But completely, can we omit the animal experimentation? Answer is no. Okay, and then we will talk about why it is like that, and we will cover details about animal experimentations also. But earlier, where I was telling you the scenario where every molecule synthesized by a chemist used to be tested in live animal, I think that has to stop, and it has completely stopped. Most of the companies don't do like that. We do a lot of in silico screening for the in-house molecules. Then we do in vitro experiments, and the compounds which are successfully clearing the in vitro screening, only highly potent compound we will take it to animal testing. Okay, so number of animal usage has reduced drastically in our country also, and CPCSA also, credit goes to them for putting the laws and very strict guidelines in place. So yes, so today's scenario, if somebody asks you how does it happen, then you can say that yes, first in silico screening, then in vivo, then in vitro in animals, and then human trials happens. Okay. On the next slide, this is just a cartoon, just why an experimental design is required. Because I hope you all will understand from this that you see, you have chimpanzee to birds to elephant to other aquatic animals to even human. We all are different in our physiology wise, anatomy wise, size and available surface area wise. So if same drug, same dose is not applicable, okay, so it has to be different. So from a lower species, when you go to higher species, there's something called as allometric scaling. So if time permits, within this 2 weeks of time, 15 days of training period, some point in time we'll also talk about allometric scaling. That if you have data available on smaller species, which is rats, mice, guinea pigs, beagle, or even monkeys, how do you actually extrapolate that data into human? That is called allometric scaling. So we will have at least half a lecture on that also, so that you can understand that how animal data is related to human data, or vice versa. Suppose you have human data available, how you can extrapolate that data back to animals, laboratory animals. So we will have a separate class for that. On the next slide, so today we are going to talk about these two cell-based models. Okay, it's called hepatocytes. Hepatocytes, so as you know, it is liver cells. Enterocyte means it comes from intestine. Okay, so within this next half an hour or so, what we will talk about is about the characterization, like their morphology, how they look like, what is generally the yield, what is the viability, what are the parameters like gene expression, and then markers for them, biomarkers, and some data, and where the future lies for hepatocytes and enterocytes is what we're going to talk about. On the next slide, so we will start with why actually you have to do this, right? Why you have to use hepatocytes or enterocytes even from human? So the answer is to have an accurate assessment of human drug properties, right? Because you see, initially you are testing in animals, but what is your final species? What is the final target species? It is human. So eventually we want to reach out to human, but we are starting with testing in animals, and even before we can do some in vitro testing, there we can use human hepatocytes and human enterocytes. Although if you want to have a comparative study, you can use rats, mice, be a dog, or even monkey hepatocytes and enterocytes along with human hepatocytes and enterocytes. Okay, so we will talk about some novel hepatic as well as novel enteric technologies. Let us explore that on the next slide. So let me spend at least a minute on this slide, because this particular slide really explains the fate of a drug. So as a student, all of you should understand, all of you are science students, and I would want that you should really understand the meaning of this slide. Okay, because see, you suffer from a cold, you take a cetirizine. You have a fever, you will be prescribed a paracetamol. Okay, or you have a pain, you will take some brufen or something. All these tablets we are taking orally. Okay, so what happens once you take it orally with water? Through esophagus it goes to your stomach, and from stomach it will reach to intestine. So your absorption can happen in stomach, or majorly it happens in intestine. So that is called absorption. So earlier we talked of ADME. You can go back to the same slide, the last slide. So Maheshwari, can you just go back to the last slide? Yeah. So once you say absorption, ADME stands for absorption, distribution, metabolism, and excretion. So this is the absorption. The pill has gone into stomach. From there, it has gone to intestine. That's where it's going to be absorbed. Where it gets absorbed? From the intestinal barrier. Okay. So there is a lipid bilayer as your intestinal barrier, and also there are some transporters, efflux transporter also are there. Okay, and then what happens is your drug will travel across through diffusion. It could be an active diffusion or passive diffusion. It crosses that intestinal barrier, and then there are small vasculatures, and I would say a portal vein is also there, which takes your drug to the liver, and that's where metabolism happens. So here, if you see, after absorption, the drug, now once it is in the blood, there will be either two forms would be there. Either your drug is there, which is in a free form. So as a student, you should know this, and please remember the drug form which is responsible to elicit the action for efficacy. It is always the free form. Okay, the free form of the drug. So where is the rest of the drug? That is bound. So where it is bound to the plasma proteins. Okay. So in blood there is plasma proteins, and to those plasma protein, a lot of this drug will be bound. That is not responsible to elicit the action. For its action, for efficacy, it is the free form which is required. So now in liver, metabolism will happen. After metabolism, elimination happens through renal. So what happens in metabolism? Majorly the CYP enzymes are involved, which will what they will do, they will try to make your molecule more polar. So the moment it is more polar, it can easily pass through, filter through kidneys. So either phase 1 reaction can happen or phase 2. If it is phase 1, there are phase 1 enzymes or phase 1 reactions, simple oxidation, reduction. This thing happens in phase 1. If it is phase 2 reactions, then what happens is to your drug molecule, there is another moiety which gets attached, like glucuronidation. So a glucuride is attached, sulfation. So like that, phase 2 reaction can happen. Eventually, it makes your molecule, drug, or a metabolite more bulky, and that it can easily pass through renal. So that is elimination. Now, through blood, it reaches to different tissues, and in tissues also they have protein. So there also drug can be as bound form, which is non-specific binding. So the free form which is available, which will go to the receptor and it will bind and it will elicit the pharmacological action. So this pharmacological action which it elicits, it is called pharmacodynamics. And on the left hand side, what all things are happening? Absorption, distribution, metabolism, excretion. This is called pharmacokinetics. So for you guys to remember this, there is a very easy one sentence definition for this. When you take any drug, what body does to the drug, okay? What body is doing to the drug, that is, it is absorbing the drug, it is distributing the drug, it is metabolizing, and body eventually excretes the drug out of your body. That is called pharmacokinetics. So what body does to the drug is pharmacokinetics, and what drug does to the body, right, which is after binding to the target, it will elicit the pharmacological action, that will be called as pharmacodynamics. Okay, so what body does to the drug is pharmacokinetics, what drug does to the body is pharmacodynamics. On the next slide. So let's jump on to our hepatocytes and enterocyte thing. So here I have just taken, this is same example, but now you can see when you take an oral dose, it reaches to through intestine. It will come out as feces, but within the intestinal barrier there could be a metabolism, because lot of cytochrome P450 enzymes which are responsible for metabolism, they are expressed in intestinal barrier. Okay, so that's why we should study the metabolism in enterocytes. That's why enterocytes are important. Suppose your drug is, after this, whatever drug is left over after metabolism, maybe it can pass through through portal vein, it reaches the liver, and in liver also there are cytochrome P450 enzymes present, so metabolism can happen there also. So liver cells also are important, that is for hepatocytes for metabolism. So you see, whatever drug is you are taking, after subjecting to metabolism by enterocyte, after metabolism from liver, hepatocytes, only drug which is available, that is bioavailable drug. Okay, in order to understand the bioavailability of a drug, you should understand the metabolism, and that is by enterocytes or by hepatocytes. On the next slide. So this is what I was talking about. If you see on the left hand side, in intestine, you have CYP. So it is the notation we use is CYP. Capital CYP is CYP. So CYP3A4. So out of all the isoforms of cytochrome P450, CYP3A4 is one of the most active cytochrome P450 enzyme. More than active, I would say. For more than 75% of the drug available in the market, they are metabolized by which enzyme? CYP3A4 enzyme. Okay, so it is the most predominantly expressed enzyme and also responsible for metabolism of more than 75% of the drug in the market. So that's why wherever 3A4 is there, chances of metabolism would be more. So they are expressed in intestine, they are also expressed in liver. So that's why we should study metabolism in enterocytes and metabolism in hepatocytes. Then there are these arrows you are seeing. So you have two kinds of transporters, again a protein, which are expressed. It could be an uptake protein or an efflux transporter, right? So based on the arrow, you can see the one which comes out, which is efflux, which gets your drug back into the intestinal lumen, and the one which helps you to pass the intestinal barrier, that is uptake transporters. Okay, next slide. So we'll start with hepatocytes. On the next slide, Please remember this will be useful to you. Hepatocytes are called as gold standards for what? For in vitro hepatic studies, drug-drug interaction studies, and hepatotoxicity studies. So, if somebody asks you that what is a gold standard to study metabolism, then it is always hepatic metabolism. Then it is always hepatocytes. And why? Because hepatocytes, for three major properties of the hepatocytes you should remember. They have intact plasma membrane. Okay, so they exhibit intact plasma membrane, and they are complete, which is uninterrupted enzymes at physiological conditions. Means they are complete cell, and there are uninterrupted levels of enzyme at a physiological condition. So what happens is it's easy to mimic this in the lab. So you can actually use hepatocytes in a lab condition, and we can really study metabolism outside of the body. So you don't have to really dose the animal or a human to study metabolism. We can study this just by having the hepatocytes in the lab. Okay, and the hepatocytes are also important. Please remember they express the real transporters, efflux or uptake transporters. So when you actually study the drugs in an in vitro environment within the lab using hepatocytes from animals or human, whatever data you will get, there is a chance that it will correlate very well with the actual in vivo data. So you will have an idea that when I dose this compound or a drug to animals or to human, what is going to be the fate of my drug from a metabolism perspective. Okay, on the next slide. So this is called as hepatocytes usage. Now, as you may understand, it's not possible to really collect hepatocytes, primary cultures from a live animal and immediately use it. Right? You just can't do it. So there is a technology available which is called cryopreservation. So cryopreservation is not a new technique. It is there since years together, from 1970s or before that it came into play. So what happens is the cells or the tissues or the organs, you're able to freeze dry them, okay, at minus 180 or 170 degree temperature in a liquid nitrogen. So what happens is it will be freezed with its properties, physiological properties intact. And then whenever you want to, so what is the advantage? Now I can cryopreserve my cells here in India. Suppose somebody wants to use same cells in US, I can simply transport it to them under controlled environmental condition. Or somebody can do the same thing in US and send it to my lab here and still use the same hepatocytes. You have to just thaw the hepatocytes. Now they will again come to normal physiological temperature. They will exhibit the same level of activity, same viability, same expression of enzymes. So I can do the experiment in my lab. So that's why you have to do the cryopreservation of hepatocytes. So what are the advantages? As I was telling you, experiments can be scheduled. If I have a cryopreserved hepatocytes with me, I can schedule my experiment that, okay, when I want to do the study, today, tomorrow, after a month, I can simply do it. But if I don't have cryopreserved hepatocytes, then I cannot schedule my experiment, because you will actually have to take a live animal, you will have to anesthetize or kill the animal, you have to perfuse the liver, collect the liver, from the liver you have to collect the cells, so primary cultures, and then you have to make sure that there is no contamination. So it's a long process. So there are commercially available cryopreserved hepatocytes, okay, and you can repeat the experiment. Exactly right. You have done the experiment. Then the other vial we have cryopreserved, that you can use it later on and repeat the experiment. So that is also one of the advantage. You can use pre-characterized hepatocytes. So now there are commercially available hepatocytes. You can choose that, okay, I want certain enzymes to be expressed more. Suppose I want a lot of hepatocyte where there is more expression of 3 or 4 enzymes. You can get such enzymes, right? Such hepatocytes. So that is possible. You can also compare multiple donors, because they are commercially available. There are single donor hepatocytes also available. There are multiple donor hepatocytes also available. So what happens is, whenever they have collected the hepatocytes from a hospital condition, wherever liver was collected, and the hepatocyte is made out of that, you can either pull them and use multiple livers pulled hepatocytes or a single hepatocyte. Okay, this is about recovery medium. So there is something called as now you have hepatocytes in a cryopreserved format, which is freeze dried. You want to use it, so you'll have to thaw and you have to recover the hepatocytes. So there is something called as UCRM. It is called as Universal Cryopreservation Recovery Media. So if you use this recovery media, you can actually thaw the cryopreserved hepatocytes. And generally, the yield for the hepatocyte is 6.28 million cells per vial. So this is how commercially they are available. There are so many vendors, and one such vendor is In Vitro Admet Labs from US, and they are the I think prevalent in the entire country. Whichever labs are using hepatocytes and enterocytes, I would say more than 75% of the labs are using In Vitro Admet Labs hepatocytes and recovery medials. So this is the yield typically you get in a vial. And what is the viability? So viability, that is number of dead cells versus the live cells. So it is up to 92% viable. Okay, so that is the viability you get. On the next slide, this is again there's something called as platable. So just for your understanding, you can have this as a suspension. So if you have it in a suspension, then you have to actually use a 96 well plate and individually do the you have to use it, or you have to use it in a small Apprentice tube. But nowadays there are platable hepatocytes are available. So even the plates are available where already platable hepatocytes are cultured for you. So you have to just get those plates. You can buy the plates and then just do the experiment and discard that plate. So it's so easy nowadays. Earlier, when I started my career, we actually used to isolate hepatocytes and enterocytes. We never used to do, but hepatocytes from the rat and mouse liver, and it was such a lengthy process. And many a times there'll be contamination, there'll be yield issue, there'll be a viability problem. But nowadays it's so easy. They are all commercially available, not that costly also. So you can actually do your experiments real quick, and you can screen your molecules for metabolism using human or animal hepatocytes. Okay. And these are the assays which you can do, like drug uptake studies, metabolic stability. Metabolic stability also we will cover in detail later. But just for understanding, suppose you take your drug, a known concentration of your drug, we incubate it with hepatocytes or microsomes. After a certain period of incubation, we quench the reaction and we take out the supernatant and then we process. So suppose we started with 100 micromolar of drug. At the end, we have left with 50 micromolar of drug. Means 50% of metabolism has happened. So that means 50% of the drug which was taken up by hepatocytes, 50% of that is now converted into metabolites, and only 50% parent molecule is left. So that will really help you to calculate the half-life of your drug. Okay, and so that's very important. So same way, if you have a platable hepatocytes, you can actually do long-term drug metabolism. Suppose you want to do a long-term, not only half an hour or 1 hour, you want to study up to 14 hours of incubation, or 14 days of incubation. Can I do it? Yes, you can do it using hepatocytes. So that is possible with a platable lots. Okay, you can do time-dependent inhibition studies. You can do enzyme induction studies. Don't worry, this may be the technical jargon words for you guys, but rest assured, by the end of 14 days, you would have learned about all this thing, at least in brief. Okay, and then the hepatocytes can also express the transporters also. So it really helps you in understanding the process of transporters, the role of transporter in viability also. On the next slide. So we have talked about hepatocytes, we have talked about in suspension format, in platable format. And this is how hepatocytes, I hope picture is, but this is really look beautiful. I have taken platable cryopreserved hepatocytes, a lot from human, from monkey, from beagle dogs, mouse, rat, and Vista rat. So this is how they look. So beautiful colony of cells you can see. On the next slide. So once you have hepatocytes in place, this is important for you that you understand where all hepatocytes can be used. Okay, and Once these hepatocytes are available, actually they are available with. Can you go to the next slide, Maheshwari? Okay. So on the next slide, what I have just shared is some data, like the lots which are available. HH means human hepatocytes, and this is the lot number. So what happens is with this human hepatocytes, actually the induction potential is already studied. So in those lot, when you try to do the induction of CYP1A2, so there are several isozymes of cytochrome P450. Like major ones you should remember: 3A4, 2D6. So one such lot
Excuse me, sir, your voice is not visible.
their data is also in place. Maheshwari,
Sir, your voice is not clear. Please check it.
my voice is not clear. You think?
Yeah, it is not clear, sir.
Okay, okay. 1 second. Just give me 1 second. Yeah, yeah. Is it any better now?
Yeah, okay.
Yeah. Yeah. Can you go back to the presentation? Yeah. All right. So this is basically induction of 1A2 using three substrates, using omeprazole, phenacetin, and another molecule which we have used is, I'm not able to see in this slide. Okay, rifampicin. So rifampicin, phenacetin, and omeprazole, and you see the induction potential for the lot. You see beautiful dose dependent induction potential you can see. See it for 2B6. All of them are showing induction. You see for 3A4, beautiful dose dependent induction. Next slide. So this is basically high viability. If I have just taken example from IVL in vitro admit labs platable hepatocytes, so they have high viability, platability, completely 100% confluent layer you can get, very responsive to all these enzymes, all the transporters they express, and long duration of cultures over 7 days can be maintained. So you can actually do quite a lot of experiments. You can screen many molecules from a single vial of hepatocytes for their metabolism liability. On the next slide. So different species, as I said, whenever you're planning to do it in your lab, you should plan for different species. Majorly rats, dogs, and human is sufficient. Even if you do only human is also fine. Next slide. So new hepatocyte tools, again, go to the next slide. There is something called as quick refreeze. Okay, so quick refreeze is like re-cryopreserve. So this is just for your understanding. This is a patented thing available, and this is like hepatocytes are cryopreserved, they are thawed, and they are re-cryopreserved successfully. So from a multiple donors thing. So that is called quick refreeze pooled cryopreserved human hepatocytes. On the next slide. So pooled cryopreserved, as I said, they are available as suspension. You can suspension. The only disadvantage is you have to use it. The moment you take out the hepatocytes, you have to use it up. Whereas with platable lots, you based on your requirement only, you have to take it out. So that is the difference between suspension versus platable. On the next slide. So what are the application? Now all of you should know. If you want to do metabolic stability, metabolite profiling, reaction phenotyping, transporter studies, inhibition induction studies, you can use hepatocytes. So don't get confused. These are very simple terms. Reaction phenotyping is nothing but, I mentioned about isoforms, right? 3A4, 2B6, 2D6, 1A2. So if you want to find out which particular isozyme, SIP enzyme, is responsible for metabolism of your drug, you can actually use that particular isoform. So that is called reaction phenotyping. Means in liver all of them are expressed. If you really want to understand which particular isozyme is metabolizing your drug, you can take that isozyme and do an experiment. It will tell you if your drug is responsible, if your drug is metabolizing by a specific isozyme. That is called reaction phenotyping, right? And if you want to see if your drug is actually inducing the expression of cytochrome P50 enzymes, those studies are called induction. If you want to see that if your drug is inhibiting the expression of enzymes in the liver, then that is called P450 inhibition studies. Okay, these are all the required studies, because when you go for regulatory submission, they'll ask for this data. Finally, you can do in vitro hepatotoxicity data also. On the next slide. So let's quickly get into enterocytes. Next slide. So we have seen hepatocytes. Here you can see in hepatocytes it is expressed 3A4. Same way it is expressed in cells of intestine. So that is why enterocytes also needs to be studied, because metabolism can happen in intestine also. Next slide. So again, why enterocytes? As all of you have understood, the first organ which is going to see the drug, if you take drug orally, is intestine, right? Before liver. In intestine only your drug is going to get absorbed. That's why if any metabolism happens in intestine, it is very important to study that. Up till nowadays, nobody was studying enterocytes, but very recently, in last couple of years, interest has increased, and now there are commercially available enterocytes available, and then you can actually study the metabolism in enterocyte. Next slide. So, as I was telling you earlier, it was not available, but primary enterocytes are now available. Again, from in vitro labs, they are available. Next slide. So, what is the morphology? Let's understand quickly how do they differ from hepatocytes. So, this is the picture of that. You can see they are very small compared to hepatocytes, but still beautiful culture you see, but they are smaller in size compared to hepatocytes. On the next slide. So this is in terms of protein it expresses. Okay, how much protein is expressed? So in human hepatocytes, it's in the range of 1.45 mg per million cells, whereas in enterocytes it's very less. How much it is? Up to 0.2 mg protein per million cells. Next slide. So let's talk about yield and viability. Next slide. So in hepatocytes, we have seen what was the yield. It was more than 6 to 7 million cells per vial and more than 90% viability. Whereas here for hepatocytes, you will generally get around 80% or 75% viability you will get, and up to 2 million cells per vial. So yield is also less and viability is also less. Next slide. So human hepatocytes, more than 75% viability, fantastic. If you get just 2 million cells per vial, that's fine. But if it is hepatocytes, then it has to be at least 6 to 7 million cells and more than 90% viability. Next slide. So these are different lots available. Like you can get all the details. You can choose which particular you want, a male hepatocytes or a female hepatocyte or enterocytes. What is the race, Caucasian, Asian, what is the age of the donor, all these things, even BMI. So based on your choice, you can choose what kind of enterocytes we want to use for our screening of molecules. As I'm saying earlier, they were not available. Now, even commercially, they were available. There are so many vendors. IVL is one such vendor who is supplying this, but you can buy from anywhere you want to. Next slide. So, drug metabolizing enzyme, uptake efflux transporters, and enterocyte markers. Very quickly, we'll see that how do you actually validate an enterocyte. Go to the next slide. So, there are cell-specific markers who are expressed in enterocytes, unlike hepatocytes, like sucrose isomaltase and maltase glucoamylase. So, Si and MAGM, these are two very specific enterocyte-specific marker genes which are expressed. So as long as you have expression of this, you are sure that you have good enterocytes with you. In hepatocytes, the marker is albumin. Next slide. So you can see here in enterocytes versus hepatocytes for these markers, you see very specific cell type markers. These are when you try to look for the hepatocytes, you will see only albumin, but it will not express these two. Whereas enterocyte will express these two, but it will not express albumin. So if two cell types are given to you, you can actually differentiate both of them. Next slide. So gene expression of particular P450. So as I said, enzymes are expressed in liver, in intestine also. Let us see the difference in that. Go to the next slide. So here, very quickly, please understand this. CYP3A4 is the major isoform which is expressed in liver as well as expressed in in intestine. So in intestine, as you can see, more than 82% is 3A4, whereas in liver, up to 40% expression is 3A4. Of course, it is major one, but more expression, where do you see? It is in intestine. And I already told you earlier that more than 75% of the drugs which are available in market, they are metabolized by 3A4. So if your drug, there is every chance that it could be metabolized by 3A4, then a better model could be intestine, right? So that's why you should study metabolism in intestine as well as metabolism in liver. Next slide. This is just the expression of different isozymes. You can see where it is available. I won't read out the entire table, but just for your understanding, 2S1, there is something called as CYP2S1. It is available, it is expressed more in enterocytes compared to hepatocytes. I mean, it is completely nil in hepatocytes. So if at all your drug is liable to this enzyme, then you should, better model would be enterocyte than hepatocytes. Next slide. So this is a paper published on that, where it proves that 2S1, which is typically expressed in intestine, but it is very less or negligibly expressed in liver. Next slide. So just recap, more than 80%, 1 to 3 million, 1 to 3 per vial, that is for enterocytes. Gene expression and marker genes we have talked about, expression we have talked about, and a specific isozyme we have talked about, which is expressed very highly in intestine. Next slide. So this is just, I just wanted to compare that to human enterocytes as well. Next slide. So I think we understood about hepatocytes. We understood about the application of hepatocytes. Now let's understand the application of enterocytes. So enterocytes is majorly used for enteric metabolic stability, right? It can be used for metabolite identification, yes, and we can do drug-drug interaction studies also. Also, since it is in intestine, even the food interaction, because food also sees your intestine first before it gets absorbed. So food drug interaction can also be studied using enterocytes. And if there is any toxicity potential called enteric toxicity, that can be also studied using enterocytes. Okay, next slide. So this is another technology on hepatocyte technology, but I think I will stop here, and we will cover remaining things, a new topic, and this remaining portion in tomorrow's lecture. I think it is close to 2 hours already. So let me just stop here. If there are any questions from students.
Sir,
Yes, Maheshwari, please.
can we stop for
Yes,
today?
yes, definitely. I'm here. So we are just
Yeah, I'm
yeah,
extremely
yeah. It's
sorry.
already 2 hours, so we'll stop here. And I think hardly 4 5 slides were there. Uh, that I'll
Yeah,
try to cover in tomorrow's presentation.
okay, sir. Students, anyone having any queries, please respond in the chat box. Sir, one student having query, I have put in a chat box. Can any one of the cell or tissues can store for more time than their life snapback?
I think answer is yes. I would say that suppose if you take a cell today, if I take my liver cells and then if we cryopreserve, I think after say after 20 years from now, or 30 years from now, I may not be alive. I'm saying forget about the cell. I may not be alive, but we can really use those cells. We can just thaw those cells and use that liver cells or enterocyte cells can be used. So that is the technique of cryopreservation. I think it's about see the point here is if you take a fresh primary culture, then the lifespan is very less, and then immediately you got to use it. You cannot even store that. So this storage technique itself is cryopreserved. So at this freeze thaw or at this quick freeze technique, what happens is at a particular stage of a cell, we are immediately freezing it at say minus 180 degrees. You're using liquid nitrogen. So what happens is all its physiological parameters or contents, everything gets freezed, and it will be in the same stage till you just thaw them. So the answer is yes, you can store them for years together, and that's the beauty of this cryopreservation technique. And I think that has made our life easy. So what is happening is now same experiment can happen in US, same experiment can happen in India because we can transport those cells, cryopreserved cells, and once it reaches here and I thaw that, I can still get the same results. Of course, with repeated thawing, the activity will go down. Yes, but at least for your first thawing, the activity, the viability, the yield should be exactly same as it was prior to cryopreservation. So, answer is yes, we can store it for many many years.
Thank you, sir. And one more question: if a drug passes in a preclinical trials, but fails in clinical
Okay.
trials, why they will do?
Okay, good one. So what happens is it's a unfortunate scenario to happen. If my drug passes in preclinical screening, in for some reason if it has to fail in clinical trial, then actually a lot of my investment will go. So generally the approach from the industry is see I cannot afford that, right? So what companies will do is as a backup they'll always have some molecules from the same series. Okay, suppose if I call it as a scaffold, I have some scaffold which is really working well for me. From that, some modification I have made, come up with one structure which works for me the best. It is giving best potency. It is safe, good metabolism, good pre-formulation data. Everything is fine. It is stable. Preclinically, it is safe. Now I take that to clinical trial, and in clinical trial, if it fails, at least I'll have some other molecules, my pipeline ready, so that I can push those molecules, of course, from the same structure with slight modification back to clinical trial. But if it has to, if specifically if I had to answer the question, if it fails in clinical trial, you can't do anything. You just can't go further with that. In fact, a lot of your investment will go away. So I would say that you should really screen them in a way that chances of failure in clinical trial are less. But it is not possible all the time. But as long as possible, I would suggest that you should really screen your molecule completely for its efficacy, for its DNPK properties, for toxicity, everything, so that chances of failure would be less when you go to clinical trial. But if at all it fails in clinical trial, then there's no other option than to drop that molecule.
Thank you, sir. Difference between Eukaryotics and anthropics.
What is that?
In the chat box, I have kept
Okay, once again. Difference
difference
between
between.
hepatocytes and enterocytes? Oh, okay. So that's what we have covered in the presentation, right? The hepatocytes means liver cells. That is called hepatocytes. Enterocytes means they come from enteric, which is intestine. So intestinal cells are called enterocytes, and hepatic cells are liver cells are called hepatocytes. There are definitely morphologically they are different. They are larger, they are smaller. Then there are gene expressions are different. The markers are different, like isomaltase and MAGMAM. MAGM is expressed only in enterocytes, whereas marker for hepatocytes is albumin. And then the expression of enzymes is also different. In CYP450, expression is different in hepatocytes versus enterocytes. Few enzyme, no, majority of the enzymes are expressed more in hepatocytes, and but few, very few are there which are only expressed in enterocytes. And the protein level is also different. In enterocytes, it is less, whereas cellular protein is more in hepatocytes. So that's a basic difference between these two. The purpose is same, although both can be utilized to study the metabolism, but morphologically they are different. They are from different tissues. They have different characteristics, and still both of them are really interesting and can really provide you a lot of important data very early in discovery.
Thank you, sir. Students, anyone having any query, please respond in the chat box. Section was very good, no queries. Yeah. Thank you,
All
sir.
right, so
Thank you
I would
so
like
much.
to thank you, all of you, and again, my sincere apologies for a few minutes of technical issue, but hopefully tomorrow we won't have such technical glitch. But yes, I hope it is interesting to you. But we have we
Sir,
talked
one more
about
question.
yeah, we have talked about introduction of drug discovery and development. Then today we have jumped into alternatives to animal experimentation. We have talked about two tools. One is enterocyte and hepatocytes. Tomorrow, of course, not this long, but very quick recap we'll do, and we'll complete this section on enterocyte, which was remaining, and then we'll take up a new topic and we'll try to cover that.
Sir, one more
Yes.
question from
So,
the
okay, I
student.
can read that. So it says, did regulatory bodies concerned about phase zero studies? Okay, interesting. So phase zero has now come up into play very recently, not recently, since quite some time. Answer is no. Okay, because see, phase zero studies are not more for regulatory; they are for us basically. For a company, or if you have a project, before doing a full-fledged phase one clinical trial, it is always good if you do with a kind of a micro sampling and with a very small population, just to understand a phase zero. So that is to build up your own confidence on your molecule. I don't think regulatory agency has any requirements from phase zero. They always look for phase one, two, three data, but they don't ask for phase zero, to best of my understanding. So that is for industry, for the scientists to gain confidence that somebody is asked that what happens if drug fails in clinical trial. So to avoid those instances, I think phase zero has come into the play. So that is more for the company rather than for the regulatory agency. Okay, so somebody has asked
Yeah, thank you, sir.
what. Yeah, one last question I'll take, and then remaining questions
Sir,
I can answer tomorrow or maybe in subsequent class. It is, what is the efficacy of hepatocytes? How much similarities it shows with human hepatocytes? So, see, efficacy of hepatocytes, I would say, see, this is, at the end of the day, it is an in vitro model. Okay. So, you cannot replace a live system with any in vitro. This is in general, I'm saying, not only for hepatocytes, for any system. You can never 100% mimic a live system with a in vitro system, right? So, but yes, as I was telling you, it is not possible every time to get into a live system to check or screen our compound. So, to ease out that process, we do this in vitro screening using hepatocytes. So now, please understand, when we are doing, it is a 2D model, basically, like 2D culture. It is, of course, nowadays 3D culture is also available. But when same thing happens in a human or an animal life system, there is a complete physiology which comes into the play. There are other organs, there are other physiological conditions, right? All those comes into the play. So there could be slight variation in the metabolism which you see in the life system compared to what you see in the in vitro system using hepatocytes. So to answer your question, how much similarities it shows, I don't think it has reached to correlation R square of 1. But yes, I would say again, it depends on compound to compound. For few compounds, it could be highly relevant from cells directly going to human experiments, but for few compounds it could be less. But in nutshell, anywhere correlation of more than zero point 0.6, I think you are better off. That's all. Even if you get a zero point 0.6 correlation, there is 60% of correlation from in vitro data to in vivo data in humans. I think you are better off. You're lucky to get even that. So please understand, the purpose of this screening is to get away with your to rank order your molecules, to rank order your compounds, that which compound we want to take it further into our screening. So only that is the purpose. Based on this, you're not going to take your final decision. So to rather than getting everything into animals, we just want to do this thing early. And sometimes I'll tell you, sometimes it happens that once you do a hepatocyte study, you come to know that there's a lot of metabolism happening. So metabolism is a liability for you. So we will go back to a chemist. He will modify the structure, and then we again test and make sure that okay, now 30% or 40% metabolism is okay. It doesn't really affect my efficacy of your molecule. So now you can proceed further. So that's where your initial cell-based metabolism will help you. Okay, but it is not a complete replacement of what happens in a live human or a live animal liver. Okay, hope I answered your question.
Yeah, thank
All
you, sir.
right. So thank you, everyone. Thanks a lot for your precious time and very interesting questions and very active participation. So looking forward to connect with you all tomorrow again at 11 o'clock. Till then, have a great day to all of you. Thank you. Bye bye.
Thank you, sir. Thank you so much. Bye bye, sir. Students, I have given the feedback form in YouTube and Facebook. Please check it and submit it. Based on that only, I will provide the certificate. Please check the feedback form and submit. I have just now posted again the feedback form.